Welcome back, everyone, to another fascinating discussion here on the Edge of Reality series, part of Beyond the Known.
Great to be diving back in.
So last time, we really went deep, didn't we? Exploring, well, consciousness itself.
Yeah, that was quite the journey. We started out there in the cosmos, looking at dark matter, dark energy.
All that unseen stuff that seems to be running the show, in a way.
Exactly. How galaxies move, how the universe is expanding faster and faster.
Yeah.
It all points to things we just can't directly detect.
And then we pivoted inward to the really tricky part, the hard problem. How does this subjective feeling, this experience I'm having right now, come from, the wet stuff in my skull?
Right. The brain, it's still such a profound puzzle. We talked about qualia, that redness of red, and just phenomenal states generally. We touched on the different camps, materialism, dualism, even those ideas like panpsychism, where maybe consciousness is more fundamental.
Yeah, a huge area of mystery, and like we said, still very much an active frontier. But today, we're making a bit of a sharp turn, aren't we?
We are indeed. We're moving from the mystery of the mind to the mechanics of the gene. We're going deep into CRISPR gene editing.
Okay, so this is about unpacking the technology itself, the advancements, the different viewpoints, getting a solid grasp on our research material here.
Exactly. Understanding what CRISPR is, how it works, and why it's become such a game changer.
All right, CRISPR. I mean, you hear the term everywhere now. So let's start at the beginning. What exactly is it?
Okay, so CRISPR, and the full name is a mouthful, clustered, regularly interspaced, short palindromic repeats. It's basically a technology that lets scientists make very precise edits to DNA sequences.
Like molecular scissors.
That's a great analogy. Highly specific molecular scissors. And the amazing thing is we didn't invent it out of thin air. It's actually a natural system found in bacteria and archaea.
Really? What do they use it for?
It's their defense mechanism, sort of like a primitive immune system against viruses, specifically bacteriophages. Those are viruses that infect bacteria. So when a bacterium gets infected by a virus, it can actually grab little snippets of the virus's DNA and store them in its own genome in these CRISPR regions. It's like creating a most wanted poster for that virus.
Your genetic memory.
Exactly. So if that same virus tries to infect it again, the bacterium transcribes those stored DNA snippets into RNA. This RNA then acts as a guide.
To find the matching viral DNA.
Precisely. It guides special proteins, the most famous one being Cas9, right to the invading viral DNA. And then Cas9 cuts it. Snip. Threat neutralized.
Wow, nature's pretty clever. Okay,
Yeah.
So how is this CRISPR approach better than, say, older gene editing methods? I remember things like ZFNs.
Right, ZFNs, zinc finger nucleuses, and talons. Good memory. Those technologies also aim to cut DNA at specific spots, but they relied on proteins recognizing the DNA sequence.
And that was harder.
Oh, yeah. You
Right.
basically had to engineer a whole new complex protein structure for every single DNA target you wanted to hit. It was laborious, time-consuming, expensive. CRISPR's big innovation is using RNA, that guide RNA molecule, to do the targeting. The Cas9 protein is sort of the constant scissor part, and you just swap out the relatively simple guide RNA to tell it where to cut. Much easier, much faster, much more adaptable.
So the guide RNA is the programmable part. How does it zero in on the right spot in, the miles of DNA in a genome?
It's that guide sequence within the RNA molecule, usually called sgRNA for single guide RNA. It's designed to be complementary to the specific 20 or so DNA bases you want to target.
Like a zipper. It just finds its match.
Kind of, yeah. It binds to the target DNA sequence through base pairing. This brings the Cas9 enzyme right to that location. Sometimes it's actually two RNA molecules working together, the cRNA and tracRNA, but often they're combined into one single guide RNA for simplicity.
And I saw something in the notes about a PM sequence. PAM, what's that about?
Yes, PM. Crucial point. PM stands for protospacer adjacent motif. It's a very short, specific DNA sequence, typically just 3 or 4 bases long, that must be located right next to the target DNA sequence that the guide RNA binds to.
So the guide RNA finds the address, but Cas9 needs this PAM sequence, like right next door, to actually make the cut.
Exactly. It's a recognition signal for the Cas9 protein itself. The most common Cas9, from Streptococcus pyogenes, SpCas9, recognizes the sequence NG, where N is any base. Without that PAM sequence in the right spot, Cas9 won't cut, even if the guide RNA is perfectly matched.
Why is that needed?
It's partly a safety mechanism, actually. It helps the bacterial system distinguish the invading viral DNA, which usually has the PAM, from the bacterium's own CRISPR locus, where the viral snippets are stored, which lacks the PAM. Prevents self-destruction, basically.
Makes sense. Okay, so we've got this bacterial immune system, cleverly repurposed.
Yeah.
But getting it to work well in our cells, eukaryotic cells, wasn't immediate, was it? What were the early challenges?
No, it definitely took some work. Adapting it from bacteria to, say, human cells involved overcoming some hurdles. One early issue was just getting the guide RNA to be stable and effective inside the eukaryotic cell environment.
It wasn't working right.
Not optimally. Researchers found they needed to engineer the single guide RNAs a bit, adding specific structures like hairpin loops, things like sgRNA plus 72 or sgRNA plus 84. These tweaks help stabilize the RNA and ensure it formed the right complex with Cas9 and the target DNA for efficient cutting in vivo.
So some molecular tinkering was needed. What was the big aha moment then?
That really came around 2012. Several groups were working on it, but the work by Jennifer Doudna and Emmanuel Arpentier and others independently showing definitively that CRISPR-Cas9 could be programmed to cut specific DNA sites in mammalian cells. That was the watershed.
That was the breakthrough.
Absolutely. It proved the concept. After that, things just exploded. People started applying it rapidly in mice, fruit flies, rats, plants, you name it. The field just took off.
Right. And since 2012, the pace hasn't slowed down, have it? The technology keeps getting better, more refined.
Not at all. It's been an incredibly fast-moving field. One big push is finding or engineering new Cas enzymes.
Beyond Cas9.
Exactly. Searching for Cas proteins from different bacteria orthologs or engineering existing ones. The goals are things like improving specificity, so they cut only where you want them to.
Reducing those off-target cuts.
Precisely. And also finding Cas proteins that recognize different PAM sequences. Cas9 needs that NG, which is common, but not everywhere. Having enzymes with different PAM requirements gives you more options for where you can target in the genome. They're even looking into more complex CRISPR systems, class 1 systems that use multiple proteins.
Expanding the toolkit, okay. And what about the type of edit? The original CRISPR just cut the DNA, right?
Yeah.
Which often just disables the gene.
Yeah, the standard Cas9 creates a double-strand break. The cell tries to repair this, often using a pathway called non-homologous end joining, or NHEJ. NHEJ is pretty efficient, but it's also kind of messy. It often introduces small insertions or deletions and dels right at the cut site. That's great if you just want to knock out a gene, disrupt its function.
But what if you want to make a specific change? Like fix a typo in the genetic code?
Oh, right. For that, you traditionally relied on another repair pathway called homology-directed repair, or HDR. If you provide a DNA template alongside the Cas9 cut, HDR can use that template to make a precise change. But HDR is often much less efficient than NHEJ, especially in cells that aren't actively dividing.
So less precise but efficient NHEJ, or more precise but less efficient HDR. That sounds like a trade-off.
It was, but that's where some really clever advancements have come in, like base editing.
Base editing, okay, what's that?
Think of it as molecular pencil, not scissors. Base editors, there are different types, like CBEs for cytosine and ABEs for adenine, and now even CGEs allow you to directly change one DNA base letter into another, C to T, or A to G, for instance.
Without cutting both DNA strands.
Exactly. That's the key. They use a nuclease dead or Nicky's version of Cas9, one that can still bind to the target DNA via the guide RNA, but either doesn't cut at all or only nicks one strand. This Cas9 is fused to a deaminase enzyme.
A deaminase.
Yeah, an enzyme that chemically modifies a specific DNA base. So the Cas9 guides the deaminase to the right spot. The deaminase makes a specific chemical change to one DNA letter, and then the cell's own repair machinery finishes the job, converting it permanently to a different letter, all without that double-strand break.
That sounds incredibly precise, just swapping one letter.
It is. A huge leap for making specific point mutation corrections. And then building on that came prime editing.
Prime editing. Even more advanced.
You could say that. It's even more versatile. Prime editing can make all sorts of changes. Single base swaps, small insertions, small deletions, combinations. Basically, you can rewrite a short stretch of DNA.
How does that work?
It uses a Cas9 nickase, so it only cuts one strand fused to a different enzyme. A reverse transcriptase.
Like the enzyme HIV uses.
Exactly like that, yeah. And it uses a special guide RNA called a PEG RNA, or prime editing guide RNA. This PEG RNA not only guides the editor to the target site, but also carries an RNA template for the desired edit.
So it brings the instructions with it.
Precisely. The Cas9, nicks one DNA strand. Then the reverse transcriptase uses the RNA template on the PEG RNA to synthesize a new DNA strand containing the edit directly at the nick site. This new strand then replaces the original sequence. It's like search and replace for DNA, but very targeted. Again, no double-strand break needed.
Wow. Base editing, prime editing, the precision is just getting incredible. But, okay, you have these amazing tools. How do you actually get them inside the right cells in a person or an animal or a plant? That delivery problem seems huge.
It is. Delivery is absolutely a critical bottleneck for many applications, especially in vivo therapies, treating someone by editing cells inside their body. There are a few main strategies being worked on. Viral vectors are a big one.
Using viruses to cure the CRISPR parts.
Yeah, viruses like AAV, adeno-associated virus, or lentiviruses, they're naturally good at getting into cells. Scientists modify them to remove the harmful viral genes and insert the genes for Cas9 and the guide RNA instead. AAV is quite popular because it tends not to cause a strong immune reaction and usually doesn't integrate into the host genome randomly.
But there are downsides.
Sure, viruses have limits on how much cargo they can carry. The gene for SpCas9 is quite large, so packaging it efficiently with the guide RNA and everything else can be tricky for some viruses like AAV. And there can still be concerns about immune responses or potential integration, depending on the virus.
Okay, so what are the alternatives? Non-viral methods.
Right. There's a lot of excitement around non-viral delivery. Lipid nanoparticles, LNPs, are a key example.
Like the ones used in some mRNA vaccines.
Exactly the same principle. These are tiny spheres made of lipids fats that can encapsulate the CRISPR components, maybe the Cas9 protein itself, or the messenger RNA that tells the cell how to make it, along with the guide RNA. They can fuse with cell membranes to deliver the cargo inside. The first in vivo human CRISPR trial actually used LNPs.
Interesting. Any other non-viral approaches?
Oh, yeah. Researchers are exploring polymeric nanoparticles, materials designed to mimic biological structures to avoid immune detection, and even using exosomes, tiny vesicles that cells naturally release engineered to carry CRISPR tools.
Trying to make it stealthy.
Pretty much. And a big focus is targeting. How do you get these particles to go only to the liver or only to specific immune cells?
Right. How do you do that?
You can decorate the surface of the virus or nanoparticle with molecules, peptides, antibodies that act like keys, only fitting locks found on the surface of your target cells. Or design nanoparticles that release their contents only under specific conditions found in the target tissue, like a certain pH level. There are even physical methods being explored, like using ultrasound or magnets to concentrate nanoparticles in a specific area.
So lots of different angles on cracking that delivery nut.
Yeah.
Okay, we've talked about cutting DNA, changing letters, rewriting sequences. But you mentioned CRISPR can do things beyond just editing the DNA code itself.
Absolutely. That's another really powerful aspect. Remember the dead Cas9, dCas9, that can bind but not cut? Well, you can attach other functional bits to it. For example, fuse dCas9 to a protein domain that activates gene expression. Then, using a guide RNA, you can send this activator to the start of a specific gene and turn it on. That's called CRISPR activation, or CRISPR.
So you can switch genes on.
Or off. Fuse dCas9 to a repressor domain, send it to a gene, and you can silence it or dial down its activity. That's CRISPR interference, or CRISPR. It's like having a programmable dimmer switch for almost any gene without permanently changing the DNA sequence, and often it's reversible.
That seems incredibly useful for research and maybe therapies too.
Definitely. And it goes beyond DNA. There are other CRISPR-associated enzymes like Cas13 that naturally target RNA instead of DNA.
Targeting RNA. What's the use case there?
Diagnostics is a big one. Systems like Sherlock use Cas13. You design a guide RNA to match, say, the RNA of a specific virus, like SARS-CoV-2. If that viral RNA is in the sample, the Cas13 gets activated and starts chopping up nearby reporter molecules, creating a detectable signal, like fluorescence. It allows for really rapid, sensitive, low-cost detection.
So CRISPR for diagnostics. Wow.
Oh.
Anything else?
One more cool application is imaging. You can attach fluorescent tags to Cas9. Then, using guide RNAs, you can light up specific locations on chromosomes inside living cells and watch how they move and organize in real time. It's like a dynamic version of older techniques like FISH.
Incredible versatility. From bacterial defense to gene editing, gene regulation, diagnostics, imaging. Okay, let's pivot to the applications people hear most about. Human health. Where is CRISPR making the biggest waves right now?
I think the most advanced and, frankly, most inspiring results so far are in treating genetic blood disorders, sickle cell disease and beta thalassemia.
Right, I've read about those trials. How does it work?
These diseases are caused by mutations in the gene for hemoglobin. The therapies generally involve taking the patient's own hematopoietic stem cells, the ones in the bone marrow that make all blood cells, editing them outside the body using CRISPR, and then transplanting them back into the patient. The edit might aim to directly correct the mutation, or, more commonly so far, to reactivate the production of fetal hemoglobin.
Fetal hemoglobin? We normally stop making that after birth.
Right, exactly. But it can substitute for the faulty adult hemoglobin. So they use CRISPR to disrupt a gene, often targeting a region called the BCL11A enhancer, which normally acts as a switch to turn off fetal hemoglobin production. Break that switch, and the cells start making fetal hemoglobin again.
And that helps with the sickle cell symptoms.
Dramatically in many cases. People like Victoria Gray, one of the first participants, have had life-changing results. It's moving towards being a potential functional cure for these conditions.
That's genuinely amazing. What other health areas are promising?
Cancer. Yes, cancer immunotherapy is a huge area. Using CRISPR to engineer patients' T cells, a type of immune cell, to make them better at finding and killing cancer cells, tailoring the immune system to fight the specific cancer. There's also early research on using CRISPR to potentially eliminate HIV from infected cells, targeting the viral DNA that integrates into our genome. Still early days, but hopeful. And then there's work across the whole spectrum. Potential treatments for inherited blindness, cystic fibrosis, Duchenne muscular dystrophy, even research into complex diseases like Alzheimer's, heart disease, obesity. The list is long.
It really sounds like it could touch almost every area of medicine eventually. What about outside of medicine? Agriculture.
Oh, absolutely. CRISPR is transforming plant and animal breeding, too. The goals are often similar to traditional breeding: higher yields, better nutrition, disease resistance.
But faster and more precise.
Much faster, much more precise. Instead of waiting generations for random mutations or crossbreeding, you can directly introduce beneficial traits or remove undesirable ones. Think crops that need less water or fewer pesticides,
Right.
or have longer shelf lives to reduce waste.
Any specific examples that are already out there?
Well, there's the slick coat cattle example. It's a natural genetic trait that helps cattle tolerate heat better. Researchers used CRISPR to introduce that specific trait into other breeds. Those have actually received regulatory approval in some places.
Wow.
And in plants, tons of work on disease resistance, making crops less vulnerable to fungi or viruses, improving nutritional value, like boosting vitamins in rice, or editing tomatoes so they ripen more slowly on the shelf.
And I saw something about biodiversity, even de-extinction.
Yeah, that's a fascinating, maybe more controversial area. The idea is you could use CRISPR to, say, make endangered plants resistant to a new disease that's wiping them out, or help native species adapt. And then there are companies like Colossal Biosciences.
Well, we mammoth people.
That's them. They're using CRISPR combined with ancient DNA sequencing to try and edit the genomes of elephants to essentially recreate mammoths, or edit pigeon relatives to bring back the dodo. It's ambitious stuff.
Ambitious is one word for it. Okay, so we've covered the science, the amazing potential in health and agriculture. But this power obviously brings up some massive ethical questions, doesn't it?
Huge questions, absolutely unavoidable. And probably the biggest one revolves around germline editing versus somatic editing.
Explain the difference there.
Somatic editing targets the regular cells in the body: liver cells, blood cells, muscle cells. Any changes made are only in that individual. They won't be passed on to their children. Most current therapeutic research focuses on somatic editing.
Okay.
Germline editing targets eggs, sperm, or embryos. The critical difference is that any changes made at this stage become part of every cell in the resulting person's body, including their own future eggs or sperm. So these changes are heritable. They get passed down through generations.
And that's where things get really complicated.
Extremely. You remember the Hejiankui incident in 2018? The Chinese scientist who announced he'd created the first gene-edited babies, attempting to make them resistant to HIV.
Yeah, that caused a massive international outcry.
Rightly so. There were huge concerns about safety, lack of transparency, questionable medical necessity, and the fact that he proceeded despite widespread calls for a moratorium on human germline editing until the safety and ethical issues were better understood. It really highlighted the profound implications of altering the human gene pool in ways that could last forever.
So safety is a major concern, even beyond the germline issue, like those off-target edits you mentioned?
Exactly. While CRISPR is precise, it's not always perfect. There's the risk of making unintended cuts or changes elsewhere in the genome off-target effects. And even on-target edits can sometimes cause unexpected problems, like large deletions or complex rearrangements of chromosomes near the cut site. Scientists are working incredibly hard to improve specificity and safety, developing high-fidelity Cas enzymes and better prediction tools, but the risks aren't zero.
Then there's the whole playinggod argument that comes up.
Yes, that's a deeply felt concern for many people, often rooted in religious or philosophical beliefs about interfering with nature or a divine plan. On the other hand, many argue that using tools like CRISPR to alleviate suffering and cure disease is a moral imperative, an extension of what medicine has always tried to do. Dr. Eric Kmenich, a prominent researcher, framed it as having the right tool and needing to focus on how to use it properly. It's a complex debate with valid points on both sides.
And related to that is the line between treatment and enhancement, right? Where does fixing a disease stop and trying to improve humans begin?
That line can get very blurry very quickly. Curing sickle cell seems clearly therapeutic, but what about editing genes associated with height, or muscle mass, or even intelligence, if that were ever possible? This leads directly into concerns about designer babies and the potential for a new kind of eugenics, where parents might try to select or engineer desirable traits in their children.
Which raises issues of fairness and equality. Who gets access to these technologies?
A huge issue. These therapies are likely to be very expensive, at least initially. Will they only be available to the wealthy, exacerbating existing health disparities between rich and poor individuals, or between developed and developing nations? Ensuring equitable access is a major societal challenge.
And what about misuse? You hear whispers about military applications.
It's definitely a concern under the umbrella of dual-use potential technology that can be used for peaceful or harmful purposes. There have been reports and discussions, some perhaps speculative, about military interest in using gene editing to create super soldiers, enhancing strength, endurance, pain tolerance, maybe even cognitive function. It sounds like science fiction, but countries are certainly exploring the possibilities. International bodies like the WHO have condemned non-therapeutic uses.
It really touches on so many sensitive areas, even how we think about disability and diversity.
Absolutely. Think about prenatal screening. We already see debates around screening for conditions like Down syndrome, some call it velvet eugenics, subtly pressuring parents towards termination. If we could easily edit out genes associated with certain conditions, like deafness, for example, what does that mean for deaf culture and identity? Who decides what counts as a disability that needs fixing versus a natural part of human variation? It forces us to confront really deep questions about what we value in society.
And even in agriculture, there are concerns beyond just the science, right? About who controls the technology.
Yes, concerns about large agribusiness companies dominating the intellectual property for gene-edited seeds. Could this lead to greater consolidation of power in the food system, making farmers more dependent and potentially worsening global inequalities? It's another layer to consider.
It seems like with every amazing potential benefit of CRISPR, there's a corresponding ethical shadow or societal challenge we need to grapple with.
That's a very good way to put it. The technology itself is incredibly powerful and holds immense promise. But how we choose to use it, the guidelines we establish, the conversations we have as a society, those are just as critical as the scientific advancements themselves.
So wrapping up this deep dive, we've gone from the bacterial origins of CRISPR to the cutting edge of base editing, prime editing, potential cures for devastating diseases, revolutions in agriculture, and right into this complex ethical maze.
It really spans an incredible range. The potential to reshape our world, for better or potentially worse, is undeniable. The research continues, focusing on making it safer, more precise, easier to deliver.
But a conversation about ethics, access, and governance needs to keep pace.
Absolutely. It requires ongoing, thoughtful public discourse involving scientists, ethicists, policymakers, and the public itself.
Which leaves us with a pretty profound final thought, doesn't it? CRISPR offers these incredible tools to potentially rewrite life itself. How do we wield that power responsibly and equitably? What are the lines we shouldn't cross? And maybe most importantly, who gets to decide where those lines are drawn in a world that's changing so fast?
That's the multi-trillion-dollar question, isn't it? There are no easy answers.
Indeed. Well, thank you for guiding us through all that complex material. It's been incredibly informative.
My pleasure. Always fascinating to discuss.
And a huge thank you to all of you listening for joining us on this exploration into the edge of reality series of Beyond the Known. If you found this deep dive valuable, please do take a moment to like, follow, and subscribe wherever you get your podcasts. It really helps us continue these journeys to the edges of understanding. We'll see you next time.